The overall goal of this procedure is to construct and validate models for non-destructive prediction of total sugar, total organic acid, and total anthocyanin content in individual blueberries by near-infrared spectroscopy.
This method will answer key problems in the field of quality evaluation of farm products such as fruits and vegetables. The main advantage of this technique is that quality of all products can be checked non-destructively by spectroscopy and the models constructed through the procedure shown here. Near-infrared spectroscopy is a powerful tool for the non-destructive quality evaluation of farm products. Prediction models are constructed by examining the correlation between the spectrum and the ingredient content determined by chemical analysis of each target. Demonstrating the procedure will be Yusuke Kimura, a graduate student from my laboratory, and Jingai, a postdoc of this facility and one of the authors of this article.
Collect a sufficient number and various types of sample blueberries from the target cultivars. Collect preferably 100 blueberries for the construction of the calibration model, and at least 10 for the validation of the constructed model. To construct robust models, collect samples of various types, such as with various colors, sizes, and at various ripening conditions. Weigh each blueberry. The weights measured are used later for the calculation of content % of ingredients of each blueberry. Next, put a blueberry sample in the center of the window of the near-infrared spectrophotometer for diffuse reflectance.